Thank you so much for having me. I'll share my screen. And
Yep, go right ahead.
so,
Perfect.
so first and foremost, thank you so much for, well, first for Devin Phillips and Chris and everyone at the MHRC for inviting me to present today. Um, yeah, I'll be presenting Breathless on Command, which is basically a play. Is that, as you'll see, the overarching hypothesis of work that we're doing in the lab is that the awareness that breathlessness or respiratory symptom perception really reflects the awareness of central respiratory motor output command. And so it was a bit of a play on words in that regard. So I'll try to recognize that it's a bit of a multidisciplinary group, and having looked back at some of the titles of presentations in the past, I didn't see much in the realm of respiratory physiology. So pardon if I do a little bit of basic principles of respiratory physiology before we kind of start presenting some of the research evidence. So this is a schematic of the neural control of the respiratory muscles and the symptom of breathlessness. So briefly, we obviously have supraspinal and spinal control, and from motor cortical and other cortical inputs, as well as pontomedullary rhythm and pattern generators, we elicit an inspiratory neural drive. Obviously, this neural drive is influenced by spinal factors, interneurons, and motor neurons that creates an efferent motor output that goes to the respiratory muscles, particularly the major muscles of inspiration, which is an active process, including the diaphragm, the scalene, the sternocleidomastoids, and various other muscles. This elicits inspiratory muscle activation, which obviously creates an inspiratory muscle shortening or a contraction and tension generation. That ultimately elicits, in inspiration at least, a decrease in intrathoracic and airway pressure to values below atmospheric pressure, creating a pressure gradient or a flow gradient. The lung will then inflate. You'll get a tidal volume or a lung volume change. And this is ultimately important because it generates pulmonary ventilation and gas exchange. Now, these actions are communicated back to the respiratory system, or the central nervous system through sensory afferent feedback from a multitude of receptors. So we've got pulmonary stretch receptors, slowly and rapidly adapting, which are actually largely inhibitory in nature. So when lung stretches, we get a progressive inhibitory signal, kind of telling inspiration to shut off. We have bronchopulmonary C fibers, J receptors, obviously muscle spindles and Golgi tendon organs. Type 3 and 4 afferents in the respiratory muscles to sense kind of proprioceptive and metaboreceptive information. And of course, very importantly, central or brain and peripheral carotid chemoreceptors. And so we kind of had this afferent-efferent relationship. In addition, there's a concept called central corollary discharge, which the muscle physiologists I'm sure are aware of, where the descending motor drive is in many ways copied to cortical limbic areas of the brain, which are the sensory areas of the brain, and can elicit a respiratory sensation or symptom perception, which I'll be referring to throughout this presentation as breathlessness or respiratory discomfort. So the basic function of the respiratory system is really to facilitate the exchange of oxygen and carbon dioxide between the environment and the body in proportion to metabolic demands. And this has implications for the regulation of acid-base balance. Of course, if we hold our breath, we become hypoxic and hypercapnic or acidotic, and that creates a feedback loop or an activation of a feedback loop to prevent us from holding our breath until we cease to exist. And so one of the factors that I think is very important for understanding, the symptom of breathlessness, and that's going to be important to today's presentation, is this concept of respiratory load compensation. And so I pardon the words, but respiratory load compensation really is that in the setting of a respiratory load, which challenges this basic function of the respiratory system, and whether that load is chemical, so that could be acidosis or hypoxia, mechanical, that could be a resistance or elastance, or muscular, so this could be weakness like neuromuscular dysfunction, or weakness due to extreme being placed under extreme loads of either intrinsic origin, so this can be disease like hyperinflation or restriction, or bronchoconstriction like in asthma, and or extrinsic origin, where you could have a corset placed around your thorax, like external thoracic restriction. And in the setting of any of these loads, the respiratory system as a whole will activate a load compensating reflex that really serves to sustain pulmonary ventilation, gas exchange, and acid-base balance by either increasing or decreasing what we'll call the inspiratory neural drive. But this has obviously implications on respiratory sensation. So this is just a bit of a guiding foundational framework for where we're going. So importantly, relevant to my lab and other labs in Canada and elsewhere, how do we glean insight? We can't really measure in any way, at least not in humans, this inspiratory neural drive, this supraspinal output. In animals, you can measure the phrenic nerve activity, for example, but we rely heavily on measures of either of the surface EMG, or in my lab and other labs, the diaphragmatic EMG. So really we can look at inspiratory muscle activation using a pretty unique catheter, this illustration courtesy of Dr. Jordan Gannett from UBC, where we place a catheter into the esophagus. It has a balloon that sits in the esophagus, another one that sits in the stomach, and these basically 10 silver electrode coils that create 5 electrode recording pairs. And what we can do is, as you can see here, the green arrows indicate inspiration, is we can look at the EMG activity of the crural diaphragm on inspiration, while we simultaneously measure changes in esophageal pressure, gastric pressure. With a pneumotac or a flow device, we can measure airflow and transdiaphragmatic pressure. So we can really look at all of these kind of actions: inspiratory muscle activation, shortening, pressure development, the change in flow, which is generating the lung volume expansion. And with a metabolic cart, we can very easily measure pulmonary ventilation and gas exchange. And the reason why we can use this diaphragm EMG as a surrogate measure, at the very least, of inspiratory neural drive is that in both humans and in animals, particularly in animals, these data come from, is that if you looked at the diaphragmatic activity versus the phrenic nerve activity with progressive increases in elastic loading in either healthy or sick animals, you can see a very, very strong correlation. So the idea here is that the EMG activity of the diaphragm is a good physiological correlate or biomarker of phrenic nerve activity, which is the neural equivalent of the central respiratory motor output command. So the overarching hypothesis for today's presentation is that respiratory discomfort, or the symptom of breathlessness, reflects the awareness of an inspiratory load compensating reflex that increases this inspiratory neural drive in direct proportion to the degree of inspiratory mechanical loading. And this reflex really acts to generate the intrathoracic pressures needed to match ventilation to the demands for gas exchange, with an overarching goal of maintaining homeostasis by stabilizing arterial as well as brain fluid hydrogen ion levels. So it's really a reflex that's trying to maintain the status quo, as any feedback related reflex is. What does all this mean? So let's take an analogy like your car. And again, if the inputs here don't align with your perception, that's fine. But let's take the analogy of a car and your cruise control. So let's say, for example, that you set your cruise control at 100 kilometers an hour. Now, this is saying akin to your pulmonary ventilation, the level of ventilation that you need to sustain in the setting of, say, a high or low load. Now, the grade of the load would be akin to the mechanical load. A flat road would be a low mechanical load, whereas a steep hill would be a high mechanical load. The engine's revolutions per minute would be akin to the inspiratory neural drive, where you would say have to have 3,500 RPM on a flat road or 6,000 RPM on a steep hill. And the fuel efficiency, I believe, is akin to breathlessness, where on a high, it's going to be high breathlessness on a flat road, or low fuel efficiency on a flat road, and it's going to be so it's going to be high fuel efficiency on a flat road, which is going to be low breathlessness, or it would be low fuel efficiency on a steep hill, which would be high breathlessness. And I always come back to this statement that this compensatory reflex, we don't get anything for free. So if you have to activate the reflex in order to sustain ventilation to maintain gas exchange and the status quo of basically acid-base balance, is that comes at a consequence. We don't really get anything for free. So if I need to increase the drive to breathe to overcome a mechanical constraint or restriction, that just comes at the expense of breathlessness. So the greater the load, the greater the compensation, the greater the symptom perception is the general idea here. So some basic principles of respiratory physiology. The first is resistance. Not to insult anybody here, but resistance obviously is the opposition to airflow caused by internal or external factors. Internal factors could be bronchoconstriction or dilation. External factors could be like wearing an N95 mask or breathing through a straw, as an example. Resistance is the relationship between the change in pressure and airflow. Airflow reflects the change in pressure required to basically overcome resistance, and volume, or lung volume or respired volume, is the airflow time integral. So let's say, for example, that we have an airway with a resistance, just for demonstration purposes, of 1 centimeter of water, generating a resistance of 1 centimeter of water per liter per second. We have a pressure gradient of 25 centimeters of water. That's going to give us an airflow of 25 liters a second. Now, if we bronchoconstrict, which would happen in an asthmatic, for example, if we didn't change the driving pressure, but we had an increase in the resistance, of course, our airflow would go down. But this is not what we want in a reflex control system. We have a demand for ventilation. So in the setting of bronchoconstriction, we would need to, therefore, increase the driving pressure or the distending pressure to ensure that the pressure gradient accommodates or takes account for the level of resistance to maintain a constant rate of airflow or ventilation. Now, of course, in bronchodilation, if you decrease the resistance, now at a given given pressure gradient, you would have twice the airflow, but you don't need excessive ventilation, for example, at a given metabolic rate. And so you can actually decrease the drive distending pressure in order to sustain a constant rate of airflow. So the general principle here is that as resistance increases, a greater difference between atmospheric pressure and intrathoracic or airway pressure is needed to generate a given rate of respired airflow and a change in volume. So in other words, to maintain a given volume, more work, the change in pressure relative to the change in volume, is needed to overcome the higher resistance, with the opposite being true if you decreased resistance. Now, another important basic principle is compliance, or its inverse, which is elastance. So here, for example, is an airway connected to, say, think of the lungs as an elastic structure like the balloon. Of course, as we progressively increase the volume in the balloon, we're going to have to progressively add or generate more pressure to overcome the elastic recoil forces of this distended balloon. And we've all probably been to a party or, where we've had to blow up balloons at a kid's birthday party. And as the balloon inflates, it gets progressively harder and harder and harder to inflate because we have to exert more effort or generate more distending pressure to overcome the inward elastic recoil of the balloon. And so the compliance is the change in volume relative to the change in pressure or the distending pressure. And again, a basic principle here is that as compliance decreases, a greater pressure gradient is needed to generate a given change in volume. Thus, to maintain a given change in volume, more work would be needed to overcome higher elastic recoil forces. So obviously in the tracheobronchial tree, we have tubes that apply kind of the resistance, and we have the lung tissue that would apply the elastance. So if we think then about pressure-volume relationships of the respiratory system, so a bit of a complicated graph here, but what I'm showing you is the pressure-volume relationship, this sigmoid S curve, which describes the y-axis variable of volume relative to pressure. So basically, this is a compliance curve. And here, residual volume, so this is when our lungs are completely empty, and here is total lung capacity when our lungs are completely full. So we're going from the bottom of our lung all the way to the top of our lung. And we have, this is our tidal volume, so at rest. And so we've got end expiratory and end inspiratory lung volumes and various other volumes. The point of this slide is that if you breathe your normal tidal breath occurs on the linear portion of this pressure volume curve, you have low elastic recoil because compliance is high, and so you have low elastic recoil. But if you breathe close to total lung capacity, where now the lungs are distended and there's an inwardly acting recoil, the elastic recoil is high. So under these conditions, when you breathe at high lung volumes, you have to generate more pressure to overcome the elastic recoil. So for example, if you were exercising from rest through to, say, moderate intensity exercise, is that the increasing demands for ventilation are, when you're breathing at high lung volumes versus low lung volumes, are going to require progressively more inspiratory as well as expiratory muscle pressure development. So in other words, the requirements for inspiratory muscle pressure development increases as lung volume and elastic recoil forces increase. Now, we also need to consider basic principles of length-tension relationships. And so this is, I'm sure, a crowd that knows this better than I do. But if we look at the relationship between relative tension and the sarcomere length, we've got this inverted U relationship where we have some optimal length where for a given basically action potential, the muscle will generate maximal tension. And as the muscles shorten, and the sarcomere length shorten, is that the relative tension, or what I'll call the functional strength of this muscle, decreases. So not only does lung volume determine the degree of elastic recoil, lung volume also is an important determinant of the functional strength of a muscle or the weakness. And this is true for the inspiratory muscles. So again, if we're breathing on the linear portion of the pressure volume curve at relatively low lung volumes versus on the higher upper A linear curve of the pressure volume curve of the respiratory system, we not only have high elastic recoil forces, but we also have low functional strength because, say, the diaphragm, when the lungs inflate, the diaphragm basically descends into the abdomen and its sarcomere shorten. So it becomes functionally weakened at higher lung volumes. So the requirements for inspiratory muscle pressure development, again, increases lung volume and inspiratory elastic recoil forces increase, but this is occurring despite the fact that there's greater functional inspiratory muscle weakness at high lung volume. So it's a bit of a double whammy. The demands are high, but the strength is low. And so the question then becomes, what are the physiological and sensory consequences of breathing against high inspiratory and or elastic loads? And perhaps it's self-evident, but at least in our field, it doesn't always seem to be. So we designed a few years ago an experiment to really start to part and parcel out the effect of lung volume and breathing pattern on measures of inspiratory neural drive in particular, which was really a way to try to look in particular at when we increase our lung volume and we have a progressively increasing inspiratory elastic loads, do we basically activate this inspiratory load compensatory reflex that's going to drive the inspiratory neural drive in direct proportion to the loading that the respiratory system, for lack of a better term, sees or experiences or feels. So I think this, again, not humbly, I'll say, I think this was a bit of a clever experiment in the way that we designed it. What we did was that we had people breathe at multiple different breathing patterns at multiple different lung volumes. So here is a spirogram where a person breathes into their end inspiratory lung volume, out to their end expiratory lung volume. And for example, we had people maintain across all breathing conditions a ventilation of about 15% of their maximum voluntary ventilation. So let's say for this hypothetical example, it's 25 liters a minute. We had everybody breathe to a metronome at 15, 35, or 50 breaths a minute, and we would adjust the tidal volume at every respiratory rate to achieve a constant level of ventilation. And so what we did was we basically had people maintain a constant ventilation at a given end inspiratory lung volume. So we had, in this case, at this end inspiratory lung volume of 65% of the inspired vital capacity, at a constant ventilation, we had a higher, basically a low frequency, high volume and a high frequency, low volume condition. And then what we did is we had people maintain the exact same ventilations and breathing patterns at voluntarily higher levels end inspiratory lung volumes. So really what this would allow us to do is to see what is the influence of where we breathe on the pressure volume curve and how we breathe at any lung volume, and also their interaction. So to bring this back to the pressure volume curve, really what we're trying to do is we were trying to get people's tidal volumes to occur on the linear and increasing to the more progressively alinear portion of the pressure volume curve. So going from high elastic recoil, low elastic recoil, and high functional strength to high elastic recoil and low functional strength. And in this case, we measured all the pressures and flows and the diaphragm EMG. And just to show you that people can, if you tell them to do something, they can do it, at least a few years ago. Here was our target end inspiratory lung volumes of 65, 75, 85, and 95. And you can see that they did very, very well at each breathing pattern in terms of achieving those lung volumes. When we asked them to breathe to a metronome at every given lung volume, you can see also that they maintained 15, 35, and 50 breaths a minute very, very well. By design, we maintained a constant ventilation at about 15% of each individual's maximum voluntary ventilation, regardless of their breathing pattern and lung volume. Of course, to achieve the same ventilation at the different breathing frequencies, at the lower breathing frequency by design, they had higher tidal volumes, and at the high breathing frequency, they had lower tidal volumes. So again, we manipulated independently and in combination breathing pattern as well as lung volume. And what we found was that, and maybe this makes perfect sense, but as the end inspiratory lung volume increased, and as people were beginning to breathe at progressively higher lung volumes where the inspiratory elastic load is going up in a dose response manner, we got a very progressive dose response relationship between the diaphragm EMG. So this is basically the diaphragm EMG or electrical activity as a percentage of its maximum voluntary value. So regardless of the breathing pattern, the drive to the diaphragm was directly proportional to the increase in end inspiratory lung volume and the intrinsic elastic loading that high lung volumes impose. So this was our first indication, perhaps, of, again, in light of numerous other studies, that there is perhaps an inspiratory load compensating reflex. Now, because people were voluntarily targeting their breathing, their tidal volumes, breathing frequencies, and operating lung volumes, we couldn't reliably evaluate somebody's symptoms. But this really was telling us that there is an interaction or a relationship between lung volume and inspiratory neural drive. Now, what we then started to do was, this is now work that my PhD student, Felix Girard, is doing. And so we spent some time and we built a kind of bespoke elastic load breathing circuit. So you can see down here, we built this variable elastic load breathing circuit, which is a series of 9 5-gallon pails, an airtight system with a repressurizing valve and so on and so forth, as well as an inspiratory loading circuit. Because the overarching hypothesis was that the respiratory system doesn't know whether or not it's having to work against a resistance or elastance. It just senses a load, right? Just like the car analogy. I set my speed, my cruise control at 100 kilometers an hour. Now to maintain that, if I can change the load, I can flatten the tires, I can put 5 Dennis Jensens in the car, so it's going to be over 1,250 pounds, or I can have the car go up a hill. Any one of those loads, the engine doesn't know whether or not it's 5 Dennis Jensens or it's a hill or it's flat tires. It just knows that it has to work against some impediment to maintaining 100 kilometers an hour. And it's going to adjust its RPMs and its fuel economy in an attempt to match that, to maintain that regulated state. So I don't think that the respiratory system knows whether it's an elastance or a resistance. It just knows that something is challenging my ability to ventilate and maintain fulfill the respiratory system's function of maintaining some homeostatic regulation, and it's going to make an adjustment accordingly. And the magnitude of that adjustment really should have some perceptual consequences. So again, we had this variable resistive load, flow resistive loading circuit, and elastic loading circuit that gets connected through some tubing to a two-way non-rebreathing valve. When we measure mouth pressures, esophageal, so we measure esophageal pressures, diaphragmatic EMG, as well as some extra diaphragmatic inspiratory muscle activities. And then we do a series of multidimensional breathlessness evaluations along the way. So and the reason why we're doing semi-recumbent cycling is actually so that people can stay completely relaxed. Jordan Gannett has demonstrated quite nicely in some previous papers that accessory muscle EMG activity is influenced heavily during upright cycling when people hold onto the handlebars. And so we wanted to do a neurophysiological study, in this case, without the influence, so people just hold their arms at their sides, relax, so we can get reliable measures of the extra diaphragmatic inspiratory muscle activity, which I won't show you in the presentation today. So Felix has been doing some really nice work on this over the last year or two. So just to orient you, there's quite a bit of data here, but first and foremost, on the left, I'm showing you the progressive elastic loading that we do, and we have people do semi-recumbent exercise, three-minute bouts of exercise at 30% of their peak power output, 50 and 70%. So we're getting progressive increases in gas exchange requirements, and at any load, we're getting progressive increases in elastance. So condition A is the unloaded control, and condition F is all the way up to 20 centimeters of water per liter loading. So that's breathing basically in two of those 5-gallon buckets. And here on the right, I'm showing you the progressive resistance. So A, again, is the unloaded condition, no resistor, leading all the way up to 17 centimeters of water per liter per second, and which we calibrated using a mechanical a lung system. I'll spare you the details. The point here is that at any given exercise intensity in elastance or resistance, you can see that, again, these are just mean values, no statistics, very preliminary results from 7 people, is that you can see that the feedback control mechanisms of the individual actually is very good at maintaining quite constant rates of ventilation. We do see a little bit of hypoventilation at the higher exercise intensities, which we might expect, but for the most part, the respiratory system fulfills its responsibility of matching ventilation to the gas exchange requirements or the work requirements in the face of progressively increasing elastic or resistive loads. Now, if we look at the inspiratory esophageal pressure, which is now the pressure that is generated by all the inspiratory muscles as a percentage of its maximum value generated during a maximum voluntary inspiratory capacity maneuver, you can see that within any given exercise intensity, so across exercise intensities, you can see that the respiratory pressures need to go up, so the respiratory muscles need to do more work, but also within any given exercise intensity, the respiratory muscle work or effort needs to increase as a function of the increased loading. And this is particularly true or visible during the resistive component. So people have maintained ventilations, but in the face of elastance or resistance, they need to generate more pressure. So again, that makes sense based on the basic principles that I was telling you about before. Now, if we look at the diaphragm EMG measures as a reflection of inspiratory neural drive, again, you can see that at any given load, there is a rise in the diaphragm muscle activation with increasing exercise intensity, as we would expect to support the higher ventilation and gas exchange requirements. But also, we see, as you can see over here, we see a dose-response relationship within any given exercise intensity, such that as we load the system to maintain a given ventilation and the need to generate more pressures, we have to activate the diaphragm muscle to a greater extent. Now, if we look at breathlessness, the hypothesis was that the increase in the inspiratory neural drive should manifest as the awareness of higher breathlessness. And again, that's what we see. So we get exercise-induced increases in breathlessness, which is not novel in any which way. But at any given exercise intensity, we can see that we get a progressive rise in breathlessness intensity ratings. So, and this is true both for elastic loading and resistive loading. Now, if we, sorry, that's the diaphragm EMG, and I just showed that. So this is the breathlessness responses rather. So you can see, again, a nice, clear kind of like step increase in breathlessness intensity ratings. When we start to look at the interrelationships between these factors, here I'm showing you I've pooled all the data. So here what I'm showing you is the relationship between the ventilation achieved at the lower dots is the 30, the intermediate dots is the 50, and the upper dots is the 70% of the peak power output. And you can see that in order to achieve any given ventilation, that when the elastance or resistance is high, individuals need to generate much higher fraction of their pressure generating capacity in order to support ventilation. In addition, they also need to activate their diaphragm muscle to a much greater extent. So again, that makes sense with basic principles. In addition, we found that for any given ventilation, with progressive elastic and resistive loading, we have a progressive rise in breathlessness intensity ratings at any given exercise intensity. But interestingly, if we look at the relationship between breathlessness and the amount of pressure that the inspiratory muscles generate, they actually are not superimposed. So in other words, although breathlessness increases as a function of increasing inspiratory pressure generation, you can see that that a given amount of pressure development, can result in very, very different levels of breathlessness. So I think that this highlights the fact that what you perceive is not the actual force output or the degree of respiratory muscle pressure. So you do not perceive the work of breathing. This is a misnomer. I think there's a lot of people out there that think that you perceive the work and the oxygen cost of breathing. That's not true. And I think that this graph shows that nicely. Very, very different levels of respiratory muscle pressure generation are occurring at a given level of breathing perception. So it's not pressure. We don't perceive pressure or the force of contraction per se. I think we perceive the degree of neural activation of the muscle needed to generate that pressure. And indeed, that's what these results show. So here I'm showing you the relationship going again from no load to high load, elastance and resistance in exercise-induced increases in breathlessness and the diaphragm EMG as an indication of inspiratory neural drive. And you can see the remarkable superimposition or overlap of these responses. So to me, this really supports the overarching hypothesis that in the face of an elastic or resistive load, it doesn't matter. They respond very similarly. In order to achieve a given ventilation to support a given degree of gas exchange and maintain the status quo, is that as the inspiratory neural, as the load goes up, the drive must go up in order to support a given ventilation and respiratory muscle pressure development requirement. So again, I think that this is quite interesting because it suggests, again, that it's not the type of load that matters. It's not the type of inspiratory mechanical load. It's just that resistive or elastic, it activates the reflex, and activation of this inspiratory load compensating reflex has a sensory consequence, in this case, breathlessness. So when I was speaking to Devin about this presentation a little bit, he encouraged me to make sure that I try to translate or make relevant these findings to our understanding of pulmonary pathophysiology and respiratory symptom burden. So I'll try to do that in my remaining slides. So here what I'm showing you is some very interesting work from Dennis O'Donnell's lab at Queen's University, where the way that they did this study is they compared the neurophysiological mechanisms, or they examined the neurophysiological mechanisms of breathlessness during exercise in people with interstitial lung disease. So these are people that basically have stiff lungs. So their mechanical limitation comes from largely an inability to expand their lungs because their total lung capacity is reduced. They also looked at people with chronic obstructive pulmonary disease. So these are people that actually have very floppy lungs. They trap gas and their lungs hyperinflate. In both cases, whether or not they have restrictive lung disease, so a low total lung capacity, or an obstructive lung disease, a high total lung capacity, the commonality of these two things is that they have a volume constraint. They basically breathe close to their total lung capacity as indicated here. So this is the inspiratory reserve volume, where 0 is total lung capacity and 2.7 in this case is breathing, or is you have the amount of room that you have available to breathe. It's the inverse, or the inverse of the end inspiratory lung volume. So as the inspiratory reserve volume declines with increasing exercise intensity, it means that the end inspiratory lung volume is rising and people are beginning to breathe on that upper alinear portion of the sigmoid pressure volume curve. Sorry, I don't know why my computer's doing this. So what you can see is that in ILD and COPD patients, they all breathe with substantially lower inspiratory reserve volumes at any given work rate compared to healthy age and sex MAC control subjects. In the setting of this, basically of these greater critical inspiratory constraints, or these greater respiratory mechanical loads, is that in order to achieve any given exercise ventilation, they must generate much more respiratory muscle activity. So you can see here at 40 liters a minute, you can see that in the controls, they're generating about 20% of their maximum EMG activity, all the way up here to greater than 50% in the COPD or the ILD. What's interesting is that the COPD and ILD people look largely indistinguishable. Two completely different pathophysiological disease states, but they're behaving very, very similarly during exercise. And indeed, when you look at the dyspnea or the breathlessness ratings with increasing exercise intensity relative to ventilation, as we know, people with COPD or ILD are much more breathless. But what's really unique about this study was that when they looked at the relationship between dyspnea or breathlessness, respiratory perception, and the drive to breathe, or the diaphragm EMG, they found that the curves across the three groups were superimposed. So again, this says that like the respiratory system of the load compensating reflex of a person with COPD or ILD, or even somebody in healthy control, they're operating the same. And it doesn't matter whether or not the constraint to breathe is coming from a restrictive deficit or an obstructive deficit. The compensation needs to be the same. We need to recruit the muscle, generate more pressure to sustain ventilation. I often say, if I have $100 bills, it doesn't matter if I take 50 off the top or 50 off the bottom. Either way, I'm left with $50. I'm $50 poorer than I was. I don't care if you take it from the top or the bottom. I've lost $50. I don't care whether it's from the top or bottom. And I think that's what this results show. It's not where the load comes from. It's just that there is a load that needs to be addressed or compensated for. So that seems to support, in some ways, the results that we showed with the elastic and the resistive load study that I showed you a few slides ago. Now, here's some work from Matt James, also from the Kingston group. This was a joint collaboration. We did a data share. And so here what I'm showing you is now within people with COPD, I'm showing you healthy control subjects in blue, and then these tertiles of people with COPD based on their degree of lung abnormality. So for simplicity, the green are people with mild lung disease. In orange, they're moderate lung disease. In red, they're severe disease. And what you can see is that the control people, if you look at another measure of volume constraint, or basically elastic loading, if you will, is the tidal volume to inspiratory capacity ratio. So this is as the tidal volume to inspiratory capacity ratio goes up, the inspiratory reserve volume goes down, or the end inspiratory lung volume goes up. Another way. It basically means that we're breathing closer to our total lung capacity where there's greater inspiratory elastic loading. And you can see that with an increasing disease severity, that people with severe COPD reach their mechanical limit at much lower levels of ventilation. It stands to reason then that if they have greater inspiratory mechanical loading at any given ventilation, they're going to require greater respiratory muscle activation. So they're going to need a greater inspiratory neural drive to drive the greater pressures needed to support ventilation. And indeed, when they looked at the measure of inspiratory restriction, the tidal volume inspiratory capacity ratio, and the drive to breathe, the diaphragm EMG activity, you can see that disease severity has no effect on these relationships. What it does is that with increasing disease severity and increasing mechanical loading, we need to generate more respiratory muscle activity or more drive to support a given ventilation. And the consequences of this is that the people with severe COPD become very, very breathless at very low VO2s or very low levels of ventilation compared to their moderate, mild, and healthy control counterparts. And if you can see it very, very clearly here. Whereas if you look at the relationship between exercise-induced increases in dyspnea on the y-axis and the drive to breathe on the x, again, in the mild, moderate, severe COPD compared to the controls, again, you can see that these relationships are largely superimposed, indicating again that as the load, as the degree of inspiratory mechanical loading needed to at a given ventilation goes up, drive goes up, and the consequence of that, again, no debts go unpaid, you become more breathless. So this idea that the body basically dissociates and there's this fundamental dysfunction and we're neuromechanically uncoupled or dissociated, I don't know why in an evolutionary system our body would do that. Like, my simple brain, I'm a pretty simple person, my simple brain is that I do more, I perceive more. Like if I'm doing an isometric bicep contraction and I start to, either fatigue my bicep or apply more load, I'm not moving, I'm maintaining the same joint angle, but I'm certainly have to recruit, either more motor units or they have to fire at different rates in order for me to maintain that. And I perceive that. So I'm not perceiving the force or the muscle action per se. I'm perceiving the input that's going in to compensate for the progressive increasing loading, either coming from an external force or the progressive fatigue of the muscle. Now, if we think of it in the opposite direction, we should be able to say then, well, if we can load the system and increase drive to achieve a given ventilation and increase breathlessness, we should be able to do the opposite. We should be able to unload the system and see the opposite effect. So here what I'm showing you, I'm the quintessential unpublished observations guy, is that if you look at the relationships here in people with COPD, if you give them a bronchodilator, so this is nebulized Combivent, it's basically a combination of two short-acting bronchodilators, very potent compared to a saline placebo, where people are doing a progressive stair-stepping exercise, is that you can see that in the placebo compared to the bronchodilator condition, breathlessness intensity ratings come down by a statistically and clinically meaningful amount. So you can see they go here from about 4 all the way down to about 2, where a minimally important difference is 1. So at a given ventilation, the symptom of breathlessness comes down. You can also see that at a given, basically in the bronchodilator condition, you can actually see now that a given drive to breathe or diaphragm EMG activity in the setting of bronchodilation results in much more ventilation. Or alternatively, achieving a given ventilation requires much less drive to breathe in the bronchodilated state. So when you remove airway resistance and you allow the lungs to deflate in people with COPD, they have to drive the muscle less to achieve a given ventilation. But interestingly, when you look at the relationship between the diaphragm EMG and the inspiratory reserve volume as the measure of where somebody breathes on the pressure volume curve, the relationships are effectively superimposed. And lo and behold, the relationship between dyspnea and drive also is largely superimposed. So this is the opposite. If you decrease a mechanical load, at a given ventilation, you need to deactivate the inspiratory load compensating reflex because you don't need to generate as much pressure and flow to support that, the pressure to support the ventilation, and that is going to be perceived more favorably. And so I'll stop here only to say that I think the current state of knowledge, which we hope we've contributed a little bit and hope to contribute some more, is that breathlessness, I don't think breathlessness reflects this dissociation or discrepancy or uncoupling of the brain to the lung axis. I really think it's a little bit more simple than that. I think breathlessness reflects the conscious awareness of an inspiratory load compensating reflex that increases the drive to breathe in direct proportion to the degree of inspiratory mechanical loading. And the respiratory system, in its wisdom or lack thereof, it doesn't care if it's resistive or elastic or both, or whether or not it's intrinsic due to disease or extrinsic due to things that we do in the lab. It just senses a load, compensates, and no debts go unpaid. And so you become breathless when things get worse, and you become less breathless if you can improve. And I think therapeutically, it means then that any therapeutic intervention capable of decreasing inspiratory neural drive. So this can be bronchodilators, supplemental oxygen. It can be exercise training, respiratory muscle training, proportional assisted ventilation. It can be anything, really. Will decrease breathlessness and respiratory symptom burden, particularly in those that are burdened by this symptom, which is a pretty large proportion of people in the community, as well, and particularly people with cardiopulmonary disease. So I think there is a common underlying mechanism. And what I hope you can take away is that if you're at a meeting and somebody stands up and says that the mechanism or the breathlessness of heart failure is different than it is a pulmonary vascular disease and different than COPD, I hope that you can think back to this presentation and say, well, maybe it's just different inputs activating the inspiratory load compensating reflex, but what breathless really is a common awareness of that activation, regardless of how you stimulate it. Again, taking dollars off the top of the pile or the bottom of the pile, you're still left with the same amount of money. So with that, I'll thank you for your time, and I'll acknowledge the people on my team that have contributed to this, particularly Felix Gerard and Frank Nero. And again, thank Devin and Chris and the MHRC for inviting me.
Thanks so much, Dennis. It's our pleasure to have you here today. So the thanks goes to you. That presentation, even though I'm in muscle metabolism, that brought me back to my cognate topics in my PhD, where I had a whole topic on cardiorespiratory control, and it literally made me think if I ever did a second PhD, this is the field I would go into. And I've said that to you, Devin. You know I've said that to you. So this was wonderful for me. I, of course, have lots of questions, but that's not my job. So I welcome questions from the audience. Please feel free to raise your hand. You're welcome to keep your video off if you want. You can turn your video on. And as always, we strongly encourage the students to ask questions. Don't be nervous. I guarantee you, if you've got a question, it's an important one. So please go ahead and ask questions, students. And faculty, if you have questions, please don't feel you have to wait for the students. Just go ahead and raise your hand. All right, Parsa, go right ahead.
Hello.
Hi.
So great presentation, Dr. Jensen. My question is going to be mainly on just PFTs, pulmonary function test. And I want to know, how do you see the role of advanced pulmonary function tests, such as DLCO, multiple nitrogen washout, and plasmography in identifying early lung dysfunctions in e-cigarette users beyond just traditional spirometry parameters?
So you're asking me that question in relation to what I just presented, or asking me that as somebody who's a respiratory physiologist?
As a respiratory physiologist.
Oh, okay. It's a little, yeah. So, I mean, I think the evidence shows relatively clearly that spirometry is a crude measure of lung function. It's not very good at identifying early disease. It's good at identifying late disease. And so I think that's certainly some work that's come out of various labs, including the Kingston lab and others, has shown quite nicely that DLCO, residual volume, and some closing volumes and things like that seem to change, or the first things that change. So they seem to be kind of good foreshadowing events of what's to come. There's some evidence that I think what's underappreciated is that the FEF 25 to 75, you can see some scooping towards the volume axis. That seems to be an early indicator of some obstructive disorders, some impulse oscillometry measures. So again, like whether or not it's due to vaping or, or environmental exposures, I don't know anything about that. I'm sure that that's the focus of your research and that's your responsibility to kind of look into. But I do know that there are more sensitive tests of early airway dysfunction than spirometry. And I would encourage, I think the best test of early airway dysfunction is when you put somebody on a bicycle and you measure their operating volumes and you put them under stress. And so if you're relying on resting tests, I think you can glean a lot more information when you put the body under stress with a controlled exercise test.
Awesome. Thank you very much.
You're welcome.
Other questions? Well, before Devin and I launch into it, no other questions right now? Okay, maybe I'll ask a question and hopefully that inspires another question from the audience. But I've got several written down. I have to pick one. Early on you show this great graph of diaphragm EMG versus phrenic activity. I think maybe it was called diaphragm activity versus phrenic activity, basically. And right away, I started to wonder how that relationship might change if there's perhaps modifications to, say, the neuromuscular junction in a disease, or if and I suppose this is part of a broader question about this interesting use of EMG as an index of the, and I wrote it down, the drive to the diaphragm is measured with diaphragm EMG measures. The drive to the diaphragm. So I kind of wondered how much of that word drive is a presynaptic concept versus the postsynaptic concept.
Yeah.
And how much can that EMG change? Do I want to use the word disproportionate to the phrenic nerve activity due to a problem at the synapse itself in a disease?
No, so I think you raise a good question, and there's a there's a bit of a debate in the literature right now. So Anna Hudson, who works with Simon Gandevia and Jane Butler, or was trained by them, she's now a PI in Australia, I think at Flinders University. She basically has talked about that what we really want to measure in terms of breathlessness understanding is the supraspinal output, right? But then that idea is that then there's the spinal motor neurons, and then it translates obviously to the muscle. So anytime that we're going to measure, say, in a hierarchy of events, a single needle recording would be the highest order, the closest basically measure, which would maybe get you there to the idea there. But if there's impairments in the neuromuscular junction or closer to, then you're getting to the diaphragm EMG and so on. And ultimately the lowest level of kind of measure would be the surface EMG. Now, to your point is that in somebody with a neuromuscular disorder or some type of impaired neuromuscular junction, you certainly wouldn't see, I'm sure that you would still see quite a linear correlation, but the correlation would be weaker. So in those studies by O'Beyer and Lorenko and stuff like that, those were studies that were done. Actually, one of them was done in an animal where they induced a septic shock, and they measured the phrenic nerve activity as well as the diaphragm EMG, and they found a very good correlation going from the healthy to the unhealthy condition with the kind of intrinsic load. But the idea there is like from the neuromuscular junction, I think what would happen is that R squared was like 0.97. I think what would happen is just that instead of the slope following along the line of identity, you would see the phrenic nerve would be intact. But if you had some type of peripheral muscle abnormality, the EMG response would probably be blunted. So I still think that you would get a linear association. It would just be that instead of it being like a close to 1-to-1 relationship, it might be like, a slope of 0.4 or 0.5, depending on the degree of the neuromuscular, dysfunction. So that's what I think. Ideally, we would love to be recording directly, say, from the phrenic nerve, but that's not necessarily reasonable. But it's interesting you raised that point, that it is a concern that, and some people actually are saying or advocating, particularly Anna Hudson, that we shouldn't even call surface or basically muscle EMG activity inspiratory neural drive. we should call it what it is, which is respiratory muscle EMG activity, and then be very cautious about it linking to that, supraspinal inspiratory neural, which is actually the inspiratory neural drive, because we don't know what happens from the spinal cord to the actual muscle.
And I can see those uncertainties coming under the spotlight in cross-sectional studies, comparing different populations,
Exactly.
or perhaps long longitudinal studies, but perhaps not in your study design, where it's the same individual breathing through different 5-gallon buckets. The odds of there being compositional changes in the neuromuscular junction in from one group to the other there is so unlikely that I guess this topic is not relevant and not likely. I don't think your data is going to be explained by changes in the neuromuscular junction between one group to the other in your design, but it could in other designs.
And I can tell you as well, like I think there's been studies by some of the groups in Italian, like Giorgio Scano's group and stuff like that, that has actually shown that I think when you do surface EMG of the peristernal, I think it is, you actually see that in people with neuromuscular diseases, like I think it's like myasthenia gravis and stuff like that. Actually, breathlessness correlates well with the EMG, the surface EMG activity.
Right.
And again, again, it's like whether or not it's a perfect correlate of the supraspinal measures, it's still going to track. So with increasing ventilation, it's still going to increase. But do you lose the direct kind of biological marker of it? It probably weakens for sure.
Yeah. And rather than being a problem, it can actually be an opportunity for a different perspective and different insight on the data, generating new hypotheses considering these things. Sometimes the assumption is the opportunity.
Yeah, and I mean also, I'm not going to get into it here, but it's an interesting conversation, is that, when you breathe at high lung volumes, there's this idea that kind of like muscle mechanical efficiency, so to speak, or like optimization, is that those supraspinal or like spinal inputs are actually going to go to the muscle that has the greatest mechanical advantage. So let's say that the diaphragm is very mechanically disadvantaged. That drive is going to preferentially go to the accessory muscles, because like, why are you going to drive a weak muscle? You're going to drive a muscle that's going to basically have, for a given input, you're going to get a relatively greater output. But again, the total overall goal is to generate a change in pressure, to achieve a given flow, to achieve a ventilation that matches gas exchange to maintain the status quo. And again, it doesn't matter. Like, so the total, we've argued, and again, I didn't present it here and I'm speaking fast, but we've argued that actually, if you just look at the diaphragm EMG, despite what I've shown today, maybe I'm shooting myself in the foot, is actually the best way to do is actually do a sum of all of the inspiratory muscles to get a global awareness. Yeah. And in the Neuro paper, we actually showed that you can actually probably misinterpret the neurophysiology of dyspnea or breathlessness, because when the diaphragm becomes very mechanically loaded at high lung volumes, it doesn't get de-recruited, but the relative recruitment, it decreases, and that of the accessory inspiratory muscles go up. So if dyspnea rises steeply, but the diaphragm is not being recruited to the same extent, towards those limits of tolerance, you could actually say it's like, oh, there's a dissociation between drive and dyspnea. It's just, you've only measured one muscle. You haven't measured the other muscles that are basically supporting the diaphragm. And if you think of it as an evolutionary system, why you wouldn't want the diaphragm to load and become fatigued and weak? Because like, you need to breathe once you're done exercising. And so the diaphragm is a muscle that you're going to protect. And I think Jordan Gannett's work has shown some nice stuff as well when they've done some pre-fatiguing of the diaphragm, and they see that the accessory muscles kick in a little bit earlier in ventilation, likely to prevent the diaphragm from really fatiguing and running the risk of not being able to ventilate when when you're not doing anything. Yeah,
wow. Okay. I see a hand up and I'll go to it, but I have to say a thank you to you because very briefly, when we measure, we'll take the diaphragm out of our mouse models and we'll measure, force frequency analysis in our Duchenne models or cancer cachexia models. And when we see force change in cancer Duchenne, I'm often left wondering if the accessory muscles have adapted and is there actually no net effect on gas, on the ability to regulate gas. So I'd love to talk to you another day about this idea of accessory muscles and how to measure them. But I'll go on to the next question here by Gazelle. Please go right ahead.
Hi there, great presentation. I just had a quick question. I was wondering if there's any scenario where dyspnea may be shifted up at a given EMG DI.
Yeah, so that's a very good question. And I mean, for the sake of presentation, I was probably being a little bit more definitive than I should have been. Certainly, I would argue, and this is something that Felix is also doing in his thesis. I think that the way that you stimulate ventilation and drive matters. I think that if you induce a very noxious stimulus, for example, like carbon dioxide, if you really perturb the homeostatic regulation of the body with exercise, is not really like it's a challenge to homeostasis, but you can maintain homeostasis. You can kind of maintain your acid-base balance throughout much of exercise. But if you have somebody breathe, say like, high levels of carbon dioxide or low levels of oxygen, becomes a much bigger threat to I think it activates the limbic and paralimbic structures to a much greater extent for a given drive to breathe. And in those cases, you would see more breathlessness for a given level of, say, respiratory muscle activation. And we didn't do that directly, but I have a paper that probably will never get cited and maybe has only ever been read by a couple of people. But it was in pregnancy, and we looked at the breathlessness response for a given ventilation, presumably for a given level of inspiratory drive, before and after pregnancy, during carbon dioxide rebreathing, as well as exercise. And the degree of breathlessness that's caused by carbon dioxide rebreathing, so central chemoreflex stimulation at a given ventilation, is 6, 7, I think it's like 4 or 5 Borg units higher. So my simple answer to your question is I think the nature of ventilatory stimulation matters. It's been not much considered. And I also think that if you are somebody with panic disorder, anxiety, you can be a hyper perceiver. So I think that there's certainly a situation where you can perceive more than your physiology. We see this clinically. We see some people that have, relatively mild disease that are just incredibly symptomatic, and we see the opposite. We see people. There's a clinical condition called near fatal asthmatics, and these people can have incredible degrees of bronchoconstriction, presumably, incredible activation of the inspiratory load compensating reflex, and they don't seek any medical attention. They don't perceive, these threat responses. So, Gazal, a simple answer to your question is yes. I definitely think that there are circumstances where you can be more breathless or perceive more respiratory symptoms for a given drive to breathe. But on the average, in the studies that have been shown, you don't see it all that often.
Okay, thank you so much for your explanation.
Very good. Any other questions? We could go to 410 if needed. Go right ahead.
Hello, Dr. Jensen. Great presentation. I just had a quick question regarding the sex differences, and that's if there's any sex differences in terms of the time point which accessory muscles get recruited.
Yeah, so I'll point you to a really, really nice study by Jordan Gannett. I think it was published, I don't know, maybe 5, 6 years ago. I forget the first author, but I know it's from Jordan Gannett's group. It's in respiratory physiology, neurobiology. And they looked at, I believe it was the diaphragm and one, if not multiple, accessory inspiratory muscles with progressive increases in exercise. And what they found was that for any given absolute level of ventilation, what you get is that you get accessory inspiratory muscle activity occurring earlier, which is to say at lower levels of ventilation in females compared to males. And so it's not a, so I mean, here, for example, I'm showing you, this is work that we did about, 10 years ago, but again, you can see that in females, here at any given ventilation, you can see that the drive to breathe is elevated and dyspnea increases in proportion. But again, Jordan Gannett's work, I think, is something that I'm familiar with, where he showed that the extra diaphragmatic inspiratory muscles become more active at lower ventilations in healthy females compared to males. And I would anticipate that response would only be exaggerated if you superimposed aging and or disease on top of that.
Thank you for the clarification.
All right, very good. Unless there's another quick question, I think this is perfect timing to please join me in thanking Dr. Dennis Jensen for this wonderful presentation and very interesting question and answer period. I I would love to keep this going to 5, but I don't think anyone else does, including Dennis. I'm going to have to reach out to you, Dennis. I've got a lot of questions. Maybe we could bring Devin in as well for these chats, but thank you so much for
You're
today.
very welcome. Thanks again for the invitation.